Hi, welcome to our next installment on the lectures on DNA chapter. Our fourth topic is what we're going to be looking at today, and that's basically how it gives directions to the cell. This is a somewhat complicated process relative to the other three topics, so we're going to break it up into two lectures. Um, okay, the big idea here is this concept of gene expression, which is really just expressing the genes that are hidden in the code of your DNA. The whole idea is to make proteins from DNA. The big idea here is that genetic information in DNA is first transcribed into mRNA and then translated into proteins by ribosomes. So if nothing else, know this big idea. This is the kind of very foundational understanding that you'll need to know in order to understand the main topic of gene expression. So let's go into a few key terms before we go into the detail of how transcription works, which is what we'll look at today. First, a gene. You guys know this. You should know this. A gene is a segment of DNA. Genes code for a specific protein to be made by the cell and then used by that cell. Proteins influence characteristics about your body that can be visible or unseen. So examples of genes would be your hair color, your blood type, any hereditary diseases you have. And then if you wanted to look at what a gene looks like in terms of its sequence of nucleotides, this might be the start of a gene. Genes can be hundreds of nucleotides long, so this is really just a brief glimpse of what it might look like. The whole point of a gene is to make proteins, which are very complex compounds made up of amino acids. Those are gonna be your building blocks of a protein. Proteins, again, they're essential to every cell process. Just to get an idea of the size and complexity of them, here we have a picture of a protein. These proteins, this model is called a ball and stick model. The balls on them stand for individual atoms or elements, and the sticks represent the bonds between them. So here is a typical protein. This is what glucose would look like. Let's expand it so you can see it a little bit. Glucose is 6 carbons, 1, 2, 3, 4, 5, 6, and a couple hydrogens, which are represented by the white balls, and then a few oxygens as well. Lastly, we have water, which is very tiny. It's 3 atoms, 2 hydrogens, and 1 oxygen. So you can see the size difference between proteins and other types of molecules that you might find. Again, here we have nucleotides are kind of the building blocks of DNA, just like amino acids are the building blocks of proteins. There are 2 main types of proteins, globular and fibrous. Globular proteins are kind of behind the scenes. They're your hormones, the enzymes that do work inside your cells, chemical messengers, membrane channels that are in the actual plasma membrane of your cells. And then fibrous proteins are things like hair, nails, structural parts to your cells. Some examples of globular enzymes, just to give you an idea of how complex and what kind of hidden stuff goes on. These are some examples. There's one called hormone-sensitive lipase, and this is basically a regulatory enzyme that splits apart triglycerides. Triglycerides are how you store your fat. So extra energy is stored as fat, and when you need to call upon that energy, if you're running a long distance race or doing something that's very physically demanding, you're gonna this hormone, this enzyme will kick into gear, and it will start making that fat more readily available to be converted into ATP. Insulin is an enzyme or is a hormone that you may be familiar with. It basically promotes the storage of glucose as a more complex molecule called glycogen. Again, you may have heard of insulin, especially with diabetics, but it is a protein hormone. Here's another one. This is a G protein coupled receptor. This protein is embedded into the membrane of the plasma of a cell, and when a hormone connects to it, like insulin, when insulin connects to it on the outside of the cell, it's going to change its shape on the inside of the cell and release another chemical messenger. So it's kind of like a tag team or something that relays a message from the outside of the cell to the inside of the cell. So those are just some examples of what globular enzymes do. Super important. They are everywhere in your body, and they're all a result of decoding genes from DNA. Oh, here's a cool little example. I don't know if it's showing up too well. This is a motor protein that's walking along a microtubule and carrying some kind of vesicle that has maybe some chemicals inside it that the cell needs elsewhere. Okay, so if our genes are in place to make proteins, why do we have to eat protein in our diet? And the answer is that there are proteins are made up of amino acids, and there are only 20 different types of amino acids that make up our proteins. Our bodies can produce 11 of them on its own. It can just take those elements, combine them, and make those amino acids. But we can't make 9 of them, so our diets have to include those 9 amino acids in what we eat. And then our digestive system will break them down and then separate them and send those amino acids to the cells that they need to be in. Okay, so here's our first major player in the process of transcription, which is that first step in decoding the code of your gene in your DNA. This enzyme is called RNA polymerase. And polymerase is basically an enzyme that creates an mRNA molecule during transcription. That's that first step. The DNA gets fed into the enzyme from this direction, and it's going to read one of the 2 strands. And as it reads that strand, it's going to connect complementary base pairs and connect them. So those nucleotides that are free floating are coming in from this channel, and they're going to connect up very momentarily just to this strand, and then it will come out on this channel, and now you have your RNA molecule. So this is basically just making a copy of the DNA code, or transcribing the code so that it can come out the other end. Some key differences between RNA and DNA. Here on the right you have DNA. It's quite obviously a double helix. Here's one of them. Here's the other strand of that double helix. RNA is a single helix. It only has one. If you compare the bases, the rungs of that ladder, you'll see that 3 of the 4 are identical. Cytosine and cytosine. They both use guanine. They both use adenine. But RNA uses uracil, where DNA uses thymine. And if you look at the molecules, they're actually very similar. The only difference is that uracil does not have a C and 3 H's, a carbon and 3 hydrogens coming off of this end of the molecule right there. There is another difference, which is why one is called deoxyribose nucleic acid and one is just called ribose nucleic acid. And here are those structural components here. If you'll notice, everything is the same about them except for this little part right here. On the sugar of a DNA molecule, there is no H. It's been deoxified, hence deoxyribose. Here the oxygen is there, and so it's just purely a ribose compound. Again, here's your standard nucleotide for mRNA. It's made up of a phosphate, a ribose sugar, and a base, which can come in one of 4 flavors: adenine, guanine, cytosine, or uracil. The M in RNA stands for messenger because it's basically carrying the message of your DNA code out into the cell. Alright, so DNA to proteins at gene expression occurs in 2 steps. This lecture is about transcription, and then the next one will be about translation. This is where DNA is transcribed to mRNA. It takes place in the nucleus. Transcription is a process in the nucleus where the genetic information in DNA is copied into a strand of mRNA. If you were to look at the complementary base pairs between one strand of DNA and its complementary strand, it's pretty simple to figure out which base pairs with the other base. ACT would bond with TGA, GTT bonds with a CAA, GAC with CTG, and AAC with TTG. RNA replaces thymine with uracil, so it's a very similar code, except that whenever you see T down here, if it's a molecule of RNA, it's going to be a U for uracil. So everything else is the same, except there's no T's. No T's, they're all replaced with U's. And that's it. That's what it means by transcription. It's essentially just a copy, but slightly different, so we call it a transcription. This video here, if you care to type it out, is actually really, really good. I can't quite get it. Let's see. I can tell you the name of it is called From DNA to Protein, and it's by Shadow Labs DO.org. But if you type in this URL address, you'll get a really nice visual of how this happens. One thing that's interesting to note, just to go into a little bit of a deeper level, is that when RNA polymerase copies this strand of DNA, it's actually going to copy the template strand, or rather not copy, but transcribe the template strand, because the template strand is complementary to the coding strand, and the coding strand is complementary to the template strand. So this blue molecule of RNA is also going to be complementary to the template strand. So if this is complementary and this is complementary, that means that this is an exact copy of this. So really what you're getting is a copy of the coding strand, with the exception that thymine has uracil. And that's basically it right there. mRNA transcript is an exact copy of the coding strand, except thymine. Instead of thymine, you have uracil. And I think that'll do it. Here's some kind of probing questions to see if you got it, got the material. What's the name of the enzyme that reads DNA? That would be RNA polymerase. What's the process called? It's called transcription. What is the name of the molecule created? That would be messenger RNA or mRNA. And what is the point? Well, the point is to get the code from DNA out from the nucleus and into the cell where it can be used. Okay, thanks for listening. If you want to hear the second part of this process, click on the next lecture. It's called translation.